all of you must be knowing it, it took off sometime in the mid 90s with the cracking of a genome, what is called as Haemophila influenza by Greg Venter's group. And if you see the data, the databases in 1996-97 period, there is only one genome was available. So that belonged to a small microorganism, that is Haemophila influenza. And if you look at the same database now, in 2019, the numbers are already displayed there. We are closer to around 3.75 lakhs of genome. So this is what is called as the explosive period in the area of genomics, right? The efforts, the data set generation, everything is in the what they call huge growth. So that is the era. So that signifies very important point for all of us. What is the use of this huge data sets? Keep on generating data is an ongoing effort, and to get the best out of the genomic data, you need to do something with the data sets. So now there is a saying, we need not sequence the genomes anymore. Rather, you start handling the sequences which is already available in public domain. So here comes the role of the so-called bioinformaticians and the application scientist. And now we talk about data scientist, artificial intelligence, machine learning. These are all reality, because who will handle this astronomical quantum of data, which is already there in a click away. So this is a snapshot of my first slide. The majority of them are microbes. Needless to say that, because they are very easy to handle and too many microbes on the earth, right? And from the pathology point of view, all major plant pathogens are already sequenced, right? So nothing left. So that does not mean that you cannot do something on genomics. You can still do a lot of genomics, getting into the data, looking at the data, and start generating the start analyzing the data for the trait of your interest. That should be your focal point. So this is a kind of efforts which is going on throughout the world. Every dot denotes there is a sequencing project which is going on. So the good part about this particular graph is these efforts are now distributed across the world. So every country in the world, they have one or other genome sequencing project. So that is a good part. If you look back at 20 years back, the genome sequencing was centered in few countries, that too in few laboratories. Only they were having the infrastructure and also expertise to do these kind of projects. Now the efforts are worldwide, and the sequencing efforts are now well represented throughout the world, and of course the maximum number of projects are centered somewhere in the United States and Europe. That is also already there. Now pathogenomics, this is not a buzzword, it is rather a very important area under microbial genomics and also under microbiology and also pathology. A lot of books are already there in the shelf. You can get into those textbooks, you can start reading what is this pathogenomics, what is its objective, what it really deals with, and how we can utilize these applications in agriculture. The rest of my talk, I will try to focus a little bit on that. And come to pathology and crop protection in particular. I am not sure how many of you really get understood or aware of the recent outbreaks of certain diseases, both in human side and also in plant side. Several outbreaks, I will just pick up a few of them maybe in the next slide. Wheat blast is one, and another one is Ebola outbreak somewhere in Africa, and a Zika outbreak in somewhere in South America. And certain things happened, and these diseases were confined by and large in those places. And here comes the role of genomics. So come to agriculture in particular, there is no doubt that the pathogens are really creating havocs. More than 15% of the pre-harvest losses are attributed to diseases. And of course, the factor which is squarely responsible for the scenario is homogeneity in crops and also intense agricultural practices. They indirectly promote or sustain the diseases by activating or by creating an opportunity for new variants of the pathogen. So, this is a reality, and invasive pathogens are the one which you need to look out, and they may create havocs in any part of the world, and some examples I will just tell in future slides. And if you compare the scenario, that is pre-genomics era and the post-genomics era, I always take this example. If you look back the history of the biology in pathology in particular, the outbreak which happened in Ireland and Europe, that is the lead blight outbreak, the mess which followed later on is mostly due to poor understanding about the pathogen, right? And the effect was lasting for more than 4 to 5 years. Story is same. From somewhere the pathogen got introduced in Europe and started creating a famine-like situation, and people started migrating, right? There is a history, and all of us witnessed it, and the world witnessed it. And the issue was there was no high-end science to understand what is really going on, right? So, if you talk about a present scenario, such a scenario may not happen. Even if it happens, we will come to know the origin or source of those infections in no time. So, that is a comfort zone we have thanks to the subject genomics, right. I will take you to the next slide. These three are standing out in the past 5 years. We have seen this in one or other part of the world, and in all the three scenarios, the tool which came into handy for the researchers, the policy makers, and also all stakeholders of this particular issue is a genomics. And this particular approach, what they termed it as field pathogenomics, right. It is nothing but the moment you cite an outbreak of a disease, you collect the sample and subject the sample to high throughput sequencing efforts. And with the kind of data sets we have, you can map all those reads and we will understand the problem in no time. This has happened in the wheat blast outbreak in Bangladesh. In a matter of a month, they almost zeroed in on the source of infection that came from a distant continent, that is South America. So, this is the potential of the genomics tools we have at hand, right. So, with this small introduction, I will take you to the proper formal presentation, that is the pathogenomics in crop improvement. I will take the case study with Magnaporthe species, in particular Magnaporthe oryzae, that is causing rice blast disease. So, myself I am A. Kumar, principal scientist in IRI, and I am dealing with genomics projects, in particular the nationally important plant pathogens. This is a scenario of the so-called rice blast. You can see wherever the stars are there, there is a report of this particular disease. Again, this is well distributed disease in all cereal growing countries in the world, in particular rice. And this is one of the major production constraints in rice, and it is caused by a pathogen belong to Ascomyces, that is Magnaporthe oryzae, right. It is a fungal pathogen. And if you want to know more about this particular pathogen, voluminous information is already available. You can get into some literatures, and a couple of things I brought it here. That this paper talks about the losses which is possible if the rice blast outbreak happens in rice fields, right. Little bit the background information about this pathogen. I am sure most of you belong to genetics and plant breeding. So, nevertheless, this is just to let you know about some of the finer details of this pathogen. This is Magnaporthe, is Ascomyces. Remember, this is one of the fast evolving plant pathogens, right, uniquely adapted in rice ecosystem, and a few other cereals, which I will let you know. The other cereals which is affected by this particular disease is pearl millet and finger millet, right? Very serious problems in those crops. And this pathogen, it evolves and evolved in agricultural fields in tropical countries where rice is cultivated in large area, right? And management options are quite strong. We have very good management options right now. The what do you call deployment of resistant genes in the form of resistant cultivars is a proven technology for rice blast management. And we also have chemical based control options, again very strong. Most of you must be knowing melanin biosynthetic inhibitors, MBI fungicides, in particular tricyclosol is a very powerful. Yes, it is a very good point that we have a host resistance and also a powerful chemical to control this disease. At the same time, these two control options are bit shaky of late. The reasons are coming from a completely unexpected quarter, that is the so called ban on the rice import which is having tricyclosol residues. This is affecting this what do you call the rice blast management altogether, because we cannot use tricyclosol any longer, and especially some countries in Europe and Middle East, they started reducing the MRL level of tricyclosol in rice consignments 100-fold. So that was a problem for us. Now that means we need to really look into an alternative approach for rice blast management in future, right? Come to host resistance, and the breeders are quite aware that this is one of the fast evolving pathogen. You develop a variety and release that variety for commercial cultivation, in a matter of a couple of years you start seeing the resistance breaking down. So the reason is this pathogen produces enormous number of conidia. The literature says that each lesion is capable of releasing 30,000 conidia. You multiply by number of plants and number of lesions if it is there, and it's really an astronomical number, and there is every possibility that a couple of cells got mutated and it really break the resistance. So what I mean to say is these two options are not really durable options for rice blast management. So that really signifies you need to understand the pathogen much deeper than what we know right now, and here comes the role of genomics, right? And another important point is the conidial germination is very quicker. In 2 hours you will see the germination happening on the rice leaf, and often forms infective presurium very rapidly. And another striking feature of this pathogen is this pathogen does not need a natural openings on the leaf to infect. That means it can forcefully enter itself into the host. So that is something which is very dangerous. So that means it's a forceful entry into the host system. So this is a little bit the background information about the rice blast. What you see here is the typical spindle shaped lesion what you see when the rice is affected, and it affects not only the leaf but also the so called the nodal. This is a nodal blast, or sometimes it is also called as the panicle blast, and it affects all stages of the crop and right up to the harvest stage, right? And we kept some biological questions before starting the project on genomics and the pathogenomics of Magnaporthe. And these are all the specific questions we kept in our mind. I think you can also keep these questions, and if you know the answer, you can tell me right away. How far the Indian blast strain is genetically closer to the strains reported elsewhere? We just wanted to know the pathogen which is causing blast in cereal crop across the world. Are they similar, or is there any difference? If there is a difference, and how far the genetic diversity is going on. So that is one question. And second question, when we started this project, the parallel time, there was a serious outbreak of blast which was happening in wheat crop in West Bengal, in Bangladesh. And naturally, it is the question we asked whether the Indian strain and the strain which is causing blast in wheat in Bangladesh, are they similar, and that is our second question. And third important question is a very tricky question. I already told that an enormous number of conidia is formed in a single field. Can we have a kind of variability in the population in a single season? So that was our third major question. The single season means you go and collect the pathogen from the single field and collect as much as possible, and you apply the best of the best genomic tools which we developed in our project, and can we find the variability or the population is clonal in the single field? That was our third major question. And of course, slightly interlinked question is the blast, which I already told, it can cause disease on leaf and also neck, whether these two stages are affected by the same strain of Magnaporthe. That was another big question, because in an open field sometimes the conflicting reports are coming. Sometimes people say that there is no leaf blast incidence, but suddenly you start seeing neck blast, and vice versa. So that was another question we kept in our mind, whether these two stages are affected by the same type of the Magnaporthe strain. That was another question. And of course, the last one is a little bit on application side. Is it possible for us to develop a kind of specific markers to differentiate the blast fungi, right? These are all the broad questions we kept in our mind, and we started addressing these questions through the genomic tools. Here comes the project on Magnaporthe genome project, and funded by ICAR under CRP Genomics. The strain what we have taken on board for genome sequencing is a strain what is written here, or MGDL, and this is a very broad host range blast pathogen, apart from originally isolated from rice, but in our experimental studies we have confirmed that it is capable of infecting other cereals also. That is already highlighted here, and it is very highly virulent pathotype. In that particular time point, it was able to knock down 24 R genes known to us. So we can see the potential of this strain. That means those R genes become ineffective in the presence of this particular strain, right? And except of course two, one is a PI54 and another one is TETUP. I hope all of you know a little bit about these acronyms and these genes, and if you do not know, I think have kind of interaction among yourself, you will understand. PI54 is a resistant gene, and TETUP is a universal donor for this rice blast. And you must be having classes on the specific genome sequencing approaches. I am not getting there. I think the specific teachers will come and handle those classes. And in nutshell, what I want to convey to you, we have used two independent approaches to sequence this genome. One is the so-called short read sequencing, that is Illumina sequencing, and another one is a long read sequencing, that is SMRT, that is single molecule real time sequencing, that is promoted by a technology what is called as PacBio. So the platform what we used here is PacBio RS2. You keep in your mind. So currently genomes can be sequenced by different approaches. The most popular approach in most of the laboratory is not a single approach, because we need two different approaches so that the genome can be sequenced in shortest possible time. This is also very, very important. You cannot prolong your genome sequencing project for months together. So nowadays this is a strategy. So thanks to the technology that really evolved now, if you talk about the historical, the yeast genome project was handled by hundreds of scientists, right? If I remember, 600 scientists were involved, 100 laboratories, and millions of rupees, dollars were invested on those kind of projects, a single fungal genome. Now, this genome sequencing project has come to individual laboratories, even up to master students thesis level. So that is what is called as the power of technology, the reach of the technology to what we are perceiving right now, right? So this is done by a small laboratory and a few individuals, right? So we have used these two technologies, HiSeq 2500 and the PacBio RS2. And these are all some of the acronyms, what do you call some of the terminologies what you normally encounter: reads, Contig, super Contig, scaffolds, and draft genome. All of you are quite familiar with this, or not familiar? Not familiar at all, right? Reads are nothing but when you subject the genome for genome sequencing, the machine reads it, correct? So, and it gives you the output. So, the length means it is a sequence length in terms of base pair. How long the machine, the technology is capable of reading, it depends upon a company to company. A short read sequencing by Illumina reads only short, 250, 300, 400 base pair, right? That is what you might have come across, 2 into 150 base pair chemistry, right? 2 into 250. Have you come across or not? Nah? The read is a kind of jargon we use here, just to tell you that the machine generated reads the raw reads which is coming from the machine. The length of those reads are squarely dependent upon the chemistry in place, right? Illumina technologies are known for giving short reads, and the other technology what is called as SMRT technology, PacBio, they are known to give long reads, right? The long read, it can be anywhere between 8,000 bases to it goes up, right? So, the advantage is short read technologies are by and large called as high accurate technologies, and long read technologies are called as high throughput technologies. So, both are equally important. You need quality and also quantity, right? And if you integrate both, the power of both technologies is integrated, and your genome sequencing project is completed in no time, right? That is why people use short read technology to address the quality issues, and long read technology to cover the genome without gap. This is one of the objectives of any genome sequencing projects, right? I will just pictorial tell you, reads are nothing but machine generate millions and millions of reads, correct? And these reads are joined and made bit longer than the machine generated one, right? This option is called what is called as Contig formation. The Contig is nothing but contiguous sequence, right? So, two reads are what they call joined. You may wonder how it is joined. They have some kind of adapters there. They know overlapping reads are there. With the help of overlapping reads, they extend the length. So, end of the day, you need to know the length, the entire genome, right? in a form what is known to exist. We believe that it is a circular form, right? You also believe, right? So, we wanted to establish what is really there in the genome, right? The order of sequences, right? What is really there in native state. We are trying to recreate in our effort. That's what the effort, the genome sequencing effort. So, the contigs are bit extended one. Then another one is super contig. So, two contigs are again enjoined and created even larger one, that is called super contigs. Then another terminology you will find scaffoldings. People use some kind of linkers to join these super contigs and make it even bigger. Scaffolds. The scaffolds are joined and made into draft genome, right? This is a kind of a sequential effort, right? It is nothing but the textbook reading, right? So, your mind perceives every character, join them and convert into word. The words are converted into sentences. Sentences are converted into paragraph. Paragraphs are converted into maybe a chapter. Chapters are converted into textbook. So, nothing more than that. So, here the only difference is it is being done by a machine, correct? So, keep in your mind these terminologies very frequently encountered in the science genomics. Okay? Contig, reads, contig, super contig, scaffolds and draft genome. I am not touched, there is one more terminology, complete genome, right. So, I have not touched it because very few genomes are really completed, right. So, the international community accept the draft genome because we know the difficulty in carrying out extra effort to complete the genome is enormous, right. Anyway, so keep these points in your mind. With that, we will just move ahead. Again, I already told how to join two reads, correct? It is not that easy like what I told. You need algorithms, you need programs, right? So, what we have used in our project is a program which accommodates reads coming from two different what do you call sequencing efforts. I already told short read and long read. So, this was a program very popular in those times, that was SPADES, St. Peter's Assembly, right. So, it is also called hybrid assembly, right. So, we use this to assembly is nothing but you are trying to reach up to the draft genome level. The entire effort is called assembly, right. Genome assembly is nothing but reads are converted into contigs, contigs are converted into super contigs are converted into what do you call scaffolds, and they are converted into draft genome, and ultimately the complete genome. The entire effort is, in one word, if you want me to summarize, it is called genome assembly. You are assembling the reads in order to get the real genome what is there in the native state. That is seldom approach, but nevertheless people are making a lot of efforts, right. Okay. So, this is assembled genome, right. So, again guidelines are there in the presentation. I will let you know what are all the guidelines and how the assembly data, what it indicates for the person who is expert on the subject, right. Just by seeing the number of scaffolds or number of contig, he will be able to judge whether you did a good assembly or bad assembly. So, these are some of the indicators. In the presentation I will let you know. Okay, once you assemble the genome, the next part is you need to really understand what is the genome composed of. You need to know what is there in the genome in the form of coding sequences, right. So, for that again you need the assistance of computer again, you need the programs, algorithms, right. So, what we have used in our project is algorithm what is called as Augustus. Augustus is a program that helps you to understand the number of genes which is present in the genome what you sequenced, right. And of course, it's a kind of routine for most of the laboratories. You need to make this data available in a public domain. So, number of public domains are available. You have GenBank, EMBL, and the DNA Data Bank of Japan, and you can put it in even specialty software databases, or the best part is you can create your own database and host it. Option is yours. Where it is after assembly, we estimated the size of this particular genome is around 35 MB, right. So, I hope you understand what you mean by 35 MB, right. 35 into 10 to the power 6 base pair, right. And we assembled the reads with the best possible algorithms what we had at the time in 996 contigs. In a colloquial or in a simple language, the 35 MB genome is represented in 996 contigs. Is it clear to all of you? The best contig, the best assembly is the one where the entire genome is assembled in one contig. So, that is the present technology what we have. Maybe some bacteria it is possible, virus of course it is possible because the length is very short, but bigger genomes it is not possible as of now, right. But still some technology offer you such a lengthy reads, so that your entire genome is assembled in 2 or 3 contigs. Now, the reports are coming. The new technology what is called as nanopore technology, it gives a really long reads, right. So, that you can assemble the entire genome in 2 or 3 contigs. So, this is also called as chromosomal level assembly, right. So, nothing better than this. If you can assemble all genome by chromosome wise, that really helps all the plant breeders to locate what is there in what chromosome, isn't it. So, that is the ultimate objective of any genome sequencing project. Technology is still evolving, maybe with passage of time we will come to we will definitely reach that stage. It's a time that matters a lot here. And of course, we published this work in a genome announcement that is from American Society for Microbiology, ASM, and this you can the title of this paper is already highlighted there. And these are all some of the take-home messages for the youngsters. The whole genome assembly with the minimal number of Contig is one of the requirement of any whole genome sequencing projects, right. The minimal number is up to you. So, if you want to for example, the flagship journal which published this genome sequencing data, what is called as genome announcement. The name is changed maybe a year ago. Now it is called as Microbial Resource announcement. They put specific conditions for the authors. They say if it is a fungal genome, you assemble your genome in less than 1000 Contigs. Very clear. If it is a bacterial genome, they tell you you assemble your genome in less than 500 Contigs. That means what? If it is excess of that, they will not accept your paper. So, you need to again sit back and again reassemble and reduce the number of Contigs and submit it again, then they will accept it. So, this is one of the conditions, right. Yeah, this is some statistics normally you encounter when you do these kind of projects. The number of scaffolds or Contig I2 996 and genome size is already given here 35 MB, and some statistics I think when you start doing it you will come to know average size of scaffolds 16,000 base pair and scaffold N50 value, maximum, minimum, and that is all about the assembly statistics. And annotation statistics, we could annotate around 12,747 genes. The average number of genes is around 1007, maximum gene length is around 140916 and the minimum is around 201. These are some statistics routinely people generate with the help of algorithms which is already there in public domain, right. And just to know little, let you know a little more about the technology per se here. We have used Illumina and PacBio. The details are given here, and every technology is known to give some quantum of data, right. That is also very important, right. So, for example, Illumina always gives you large volumes of data. For example, here in this our own project, we could generate around 10 GB of data from one strain, that is RNGTL. And PacBio, it gives a less quantum data but long reads. So, we could generate around 500 MB data in one SMRT gel we could generate, right. And this is the front page of Spades, that is assembly which accommodates reads from competing companies, right. This is Augustus, the front page, what is called as the gene prediction algorithms. So, here also options are yours. There are two kinds of prediction. One is what is called as the reference based prediction, and that is pretty easy. If the genome is already sequenced, the information is already available, people would not invest too much time on what is called as a de novo annotation. They go for reference based annotation. That is what exactly we did, and the reference what we used is Saccharomyces cerevisiae, into the assembled genome. So, the statistics is already highlighted here, and these are all some of the routine. Once you annotate, then the next question comes, what this genome is capable of doing, right? What is called as the pathway analysis, right. So, use again softwares and programs which is available. The very popular one, I think most of you may be knowing, the KEGG and automatic annotation server. It is a routine kind of programs normally people resort to, and this we have done it, and these are all some of the statistics. The output of such an analysis you will get under different headings, for example, metabolism related, genetic information processing related, environmental information processing related, cellular process, and organismal systems. So, it is a kind of blanket data one can expect from any genome sequencing projects, right. This is very important for pathology. students and pathology researchers, because this is one specialty database which searchable option, PHI base. This is also called pathogen host interaction base. What it does is it will tell you the pathogenicity and virulence related genes in your genome. So, the input data you give, and it identifies the annotated gene is capable of doing what. For example, whether it is having virulence function, effector function, whether it is having pathogenicity, hypervirulence, or whatnot. So, this is very useful, and you can at least form some opinion about the kind of data what you generate. And this is another offshoot data people normally generate, whoever is involved in microbial genome sequencing projects. This is what is called as identification of transposon coding sequences in the genome, right. So, transposon PSI, an application of PSI, PSI BLAST to mine the transposon over of homologies in the genome. So, what we found is in any genome, microbial genome, whether it is a fungal or bacterial genome, you see a lot of repetitive transposons in the genome which is distributed throughout the genome. So, if you want to know about them, use this one. After having generated the genome data, you subject the genomic data for transposon PSI analysis. That gives you how many transposons are there in your genome. So, you can just read it here, different classes of transposon and also how many copies they are there in the genome. This is also very useful because we know that transposon play an important role in evolution, right in the, I already talked about pathogenic variability which is happening in a pathogen like Magnaporthe, and we cannot underplay the role of these transposons in making a new variants available in the population, because their ability to hop on and hop off in the genome, that quickly creates a variability in the virulence coding genes and come out with a kind of new type of pathotypes, right. Yeah, this is the place where you can deposit your genomes, and it involves a lot of series of interlinked steps. You need to work with the database curators, and you have to submit the data. They will verify, quality check. It's a kind of a series of efforts are there. The moment your data is uploaded, that only indicates that you have done a fairly good job in terms of generating genome sequencing data. This is our genome. We what we call hosted in GenBank. That was all about the RISE BLAST genome sequencing effort I just shared with you, and I already told we have the BLAST in other cereals also in our country. In particular, I would like to draw your attention to millets. Pure millet is one crop which is severely affected by this the BLAST disease. Right, you are right here, and right in the field we have pure millet, and some genetic students are sitting here. You take their help. If you really want to see the severity of BLAST in pure millet, just go and see. No plant is free from this BLAST lesion in our field. So, you can have a look. Excuse me. So, BLAST is not only serious production constraint in rice, but also in many other cereals, and in particular millets, finger millet, small millet and pearl millet are severely affected, right. This is a typical pearl millet BLAST, and we have selected one of those pearl millet strain for genome sequencing, right. The reason is when we started this project, there was no information about pearl millet BLAST infecting strain, particularly the genomic data. It was not there. Excuse me. And we published in Microbial Resource, Microbiology Resource announcement. It's the first draft genome sequence of pearl millet BLAST pathogen Magnaporthe griseae, strain PMG-DL, obtained using PacBio single molecule real time and Illumina NextSeq 500 sequencing. Again, the strategy is same. We adopted long read and also short read, right. I do not want to again get into the discussion. The primary motivation, which I already told, there was no data available, and we kept some of the questions in our mind. This is a strain what we used, PMGTL. Again the story is same, two different approaches, a short read Illumina and a long read PacBio, right. Genome assembly was done using anybody? Spades, yeah, good. This is the data sets what we generated. There is a small intervention here. The Illumina technology, by the time we reached last year, the Illumina technology also got improved, right. So, if you remember the previous one, it was HiSeq and here it is NextSeq, right. That was a big difference. NextSeq 500. Please note this finer difference, right. When we started the previous one, it was HiSeq 2500, right, and this was done using another chemistry, that is NextSeq 500. Why I am highlighting this, you will just come to know in terms of data quality. So, here we could generate 3 independent reads, one from PacBio of course and another 2 from Illumina. One is what is called as Paden library, another one is MatePair library. So, I hope you would read in the coming days the difference between Paden library and MatePair library. These are all interlinked, but again very distinct approaches for sequencing whole genome, right. So, just look at the data, the quantum of data what we generated. Paden library gave us around 13 GB data and MatePair gave us around 3.4 GB data and PacBio gave us around 1.1 GB data, right. We have so much of data generated from one genome. Now, the next thing is you need to assemble, right. So, we did that, right. So,
73x means we have tried to 73 times.
we will have some discussion on that. Okay. Yeah. That is total, what she is asking is what is the meaning of 273x, which is given in the last coverage, 71x and 23x. So, this is nothing but estimated genome size and also the data what you generated, and the total data what you generated, the total data what you call total basis divided by the estimated genome size would indicate how many times each read is sequenced, depth of sequencing, right. This is 273x means a particular read is sequenced that many times. Okay. So, this as simple as that. More coverage means better accuracy, correct. If you sequence only once, there may be a possibility that some errors are already loaded there. How do you rule out that? You need certain X coverage of your genome sequenced, right. Then what is called as you need to really spend time with this small things. Then they will take, you have a 273 repetition of the same read and it will read all of them, and whether all the bases are in consensus with each other. If all of them are consensus with each other, what is the meaning of it? It is very high degree of accuracy, right. Even if some errors are coming, because machines can make errors. In the same base call, out of 273, couple of them are giving you some other read, some other base, then it goes with the majority, what is called as consensus average, correct. These are all some of the terminologies we use. And the best fit, you must have studied in statistics, right, the best fit, right. So, this is so important, the coverage, and usually this Illumina reads always gives you very high depth coverage, and PacBio will gives you a low coverage, but long reads would be there. Okay. It is good that you asked that question, right. So, this is some of the assembly statistics. Here comes the big difference. In the previous genome, it was 996 context, but you just look at here, we could assemble the genome in 341 scaffolds, right. Genome size is around 48 MB, right. Just imagine the genome is larger than the previous one, and we could assemble in less number of contact. So, in all standard, this assembly is marginally better than the previous assembly, right. So, 341 scaffolds, they represent 47.89 MB of genome, right. And again, average scaffold size is here, and 50, all those routine things are highlighted here. So, again remember, Augustus was the program used for what? Gene prediction. You can use reference based gene prediction, okay. And these are all some of the statistics, so no consequences for you. The message is, you remember this name Augustus, it is a gene prediction program. You can use the reference based gene prediction using this particular. Again, PHI database for what? Anybody? Host pathogen interaction or the interactome analysis, the pathogen side, what is really contributing towards their virulence and pathogenicity, you will come to know using this particular program. In this particular genome, we found around 850 CDS genes. They have role directly or indirectly on pathogenicity, virulence and effector function, right. And of course, these are all routine KEGG analysis people do, and it comes out with the different data sets. For instance, in this case, the number of genes which is having role in biological process around 4000 CDS, and cellular components around 3356 CDS, and molecular function is around 4174 CDS. These are routine output you normally get from such a work, right. And this is 3151 genes were annotated into 24 functional categories, and that is highlighted here. Just skip this kind of slides, I will come to the main one later. And this particular data is available in public domain with the number what is already highlighted here. Yeah, all these data sets were already uploaded in a public databases, anybody can access from anywhere. Yeah, this is a strain, the details. The good point is, some take home message from our own effort. The pale millet infecting Magnaporthe gracia was genome sequenced and using Illumina and PacBio sequencing platforms, and to the best of our knowledge, this is the first genome sequencing data of pale millet infecting blast fungus. And of course, the hybrid assembly generated or revealed rather 48 MB of genome, and we could annotate around 10,000 genes, and the genome was already published. This is a comparative statement between these two genomes, one infecting rice and another one infecting pale millet. So, which I already highlighted here in the previous. So, the take home message is the rice genome is smaller than the pale millet genome. Pale millet rice infecting Magnaporthe genome is smaller than the pale millet infecting Magnaporthe genome, right. So, one is around 35 MB and another one is 48 MB roughly. So, some of the statistics are already highlighted here. Yeah, this is the key point which I already highlighted here, right. If you do not have any prior information about any genome, suppose if you want to start a genome sequencing project, right, and you are not sure whether somebody has already done it or not, or it is going on or not. So, how to do that? Google it out, fine, as simple as that, isn't it? Anything can be searched in Google, genomes are no exceptions, right? But we have a very specialty database, what is called GOLD. GOLD stands for Genomes Online Database, right, very user friendly, and it update on a regular basis, and you will come to know whether the genome of your interest is already sequenced or it is going on in some laboratory. It will give all the details of the ongoing genome sequencing projects. That is the beauty of this particular database. So, the figure what I showed, this the first slide is from here only, right, and it is regularly updated. okay, and you have lot of options are there. Remember this, right? So when I had a quick look at the Magnaporthe genomes, how many of them are really going on. If you go back 2004, only one genome was sequenced, and that paper appeared in Nature, right? So now it's not possible. So, right? So rather impossible, right? So now hundreds of strains are already sequenced. The number is always going up, right? Every passing week, somebody has already started a genome sequencing project, and they put it here. This is so important for us because this would avoid a potential duplication of efforts. Please remember, just because you have some decent fund, it does not mean that you have to do this work, right? Unless the situation warrants. Don't get into genome sequencing efforts, right? Maybe the required information, what you keep in your mind, or the biological question, what you keep in your mind, may be addressed with the data set which is already available. So why do you want to generate the genome sequencing data at all, right? This is one question. So read some of the papers appeared, the do's and don'ts for genome sequencing project. In my opinion, I think you should make a thorough literature survey and understand if it is already there. Let's not do that. Rather, you invest your time and money on analyzing the data which is already available. I think that would be the best strategy to reach the target questions or whatever you have in your mind. Okay? This is a snapshot of the genome sequencing in one organism, that is Magnaporthe, hundreds of sequences. Remember, this is the one people were conscious enough to put it in this database. This is not speaking of those people who is not putting their data here, right, or putting their information here, right. That means the number is always more only, right. It's a single window system in the sense with the cooperation of all the workers, this database is running, right. Yeah. So that was all about how we approached the genome sequencing project. One is the rice infecting Magnaporthe, another one is fuel millet infecting Magnaporthe. I hope I conveyed something to you. The rest of the presentations, I would focus on how to utilize the sequence data for the biological questions, whatever I raised in the beginning of the presentation, right. I will take one by one. The first one is genome-wide comparison between fuel millet infecting Magnaporthe and rice and finger millet infecting Magnaporthe. This is our question, right. So you'll appreciate maybe after these few slides why we have started this work. This is something like utilization of whole genome data for comparative genomics of Magnaporthe species. And of course, the questions we kept in our mind, one is whether the strain which is causing rice blast and fuel millet blast in our country, whether it is similar to the one which is causing blast in wheat in the neighboring country or even in South America. That was one question. Another question is the intra-field population collected in a single season, is there any variation? Or the leaf and neck blast, whether are they divergent population or they what do you call the same duplicate population. These are all some of the questions, right. We have standardized, or rather we wanted to develop a new tool for population genetic analysis of Magnaporthe using the genome data. That was our second question. So what we have done, we have adopted a tool that was in the hands of a medical microbiologist, what is called as multi-locus sequence typing, right. It was a very important tool. People use this tool to track the movement of potentially dangerous pathogens across the continents and across the nations. So this is we have adopted this tool, and we generated some data from our own data sets, right, and we have used several isolates in the report. Question number one, how far the Indian blast strain is genetically close to the strains reported elsewhere. The assembled genome was compared with the 15 other genome of Magnaporthe rice. Remember, I already told hundreds of genomes are already uploaded. Remember, all those genomes need not be having high quality assembly. This is one of the challenges, right. High quality assembly means the draft genome with less number of Contigs. So, at that particular point of time 2017, we could find around 15 genomes with a decent statistics and what do you call a good data set. We have taken all of them on board, right, including the genome of the Brazilian wheat blast isolates. Okay. This is again the field pathogenomics I talked about. So, what was done in Bangladesh, they created a kind of open source or what is called open wheat blast platform, where the data is instantly uploaded. No data was kept in the confines of the laboratory. Whatever data generated, genomic data, it was just hosted in no time. That means the data was shared with all. That was a what do you call a single decision which really helped all the researchers, right. So, we had access to this data, right. So, this is a kind of the phylogeny we just found, the 15 whole genome and the Indian rice blast genome. Using the whole genome data, the phylogeny was created. So, roughly we could see around 4 clusters. Are you seeing the 4 clusters here? Right? If you look at the cluster number 1, all of them are South American wheat isolates, wheat blast isolates, correct? Do not take wheat isolates, it's a wheat blast isolates. The second cluster is all about again wheat blast isolate and also ryegrass isolate. Remember, the ryegrass isolate is capable of infecting wheat also, right? And third one is very interesting, that is a grass isolate. And the fourth one is really interesting, all our Indian isolates. The one which is highlighted as AK1 is the RMGTL, the genome what we sequenced, right? At that particular year, we could see 3 other genomes, MG01, B157 and 7015. 7015 was the original strain sequenced in 2004. The paper appeared in Nature, we already told you that was this strain 7050. It's a very famous strain, now it is treated as one of the type strain for genome sequence data, right. A chromosome assembly is there and all data sets are nicely annotated, and we refer this one for all practical purpose, right. So, all these rice infecting blast isolates, they clustered together. So, that is a beauty of this particular work what we found. Remember, irrespective of what do you call the origin of those isolates, except 3, AK1, MG01 and B157, the rest all the last one is coming from United States, and it again clustered together, right. So, it indicates somehow the host is clearly coming out as a kind of a factor which we cannot ignore, right. This is a second one what is called as the comparison between our isolate with the one which created a blast epidemic in Bangladesh. So, here again what we found was it was genetically distinct. The one which is causing rice blast in our country is completely distinct from the one which is causing wheat blast. That means it is unique, our strain is unique, and that strain is also very unique. That is also has come out from this particular work. And of course, the statement is not my statement, I just borrowed from some literature. The agronomic consequences of the long distance pathogen migration are currently unpredictable, right. So, wheat blast was one off incident, it created a havoc in 15,000 hectares, right. Similar migration is always happening in all other plant and all other crop and all other pathogens as well, but very few outbreaks we are witnessing. That is what the statement is, highly unpredictable. So many other factors would play a role here, genetic homogeneity of the host, no doubt, right, and the environmental conditions which is favorable for the disease to come out. These are all some of the factors will influence, right. But the second statement is very important. The pathogen population may not pose a significant threat to the crop production in the country of the origin. For example, the South American continent, still they are living with this rice blast, their production. Of course, it might have been suffered a little bit, but they could sustain the yield. Right. When it come to another country, it created havoc. So, this is what is called as transboundary migration of pathogen is always a threat, and it can really create a devastating effect in the country where it finally lands. Right. From this perspective, sharing of the information is so important. That is what it happened, and all genome data was shared with Rustall, and quickly people concluded from where this pathogen has entered. Right. This is a statement that we made. The Indian strain of Magnaporthe infecting rice is genetically distinct from the one which caused blast epidemics in South America and also Bangladesh. So, we could answer at least the one question what we kept in our mind. The second question is intrafield. I will take another 15 minutes. Somebody is waiting. Yeah. Okay, I will continue. The genetic variability in intrafield. So, this is another very tricky question we just wanted to answer, and again the link between leaf and neck blast. So, here what we have done, we have just selected some genomic region. In total 18 genomic region we have selected, and 11 of them are having a role in housekeeping functions in Magnaporthe, and 7 of them are having virulence function in Magnaporthe. So, total 18. And the population what we handled is came from a single field located in Srinagar in Kashmir. So, around 22 we received, and with our own collections, 24 population we handled in this project. And we included deliberately a finger millet and also a pale millet Magnaporthe also in the whole project, right. And the flow is something like this. The primers were designed and the amplification sequencing conditions were standardized. Sequence reads were generated from each of those loci coming from 24 isolates. Okay. You have 24 isolates, and each isolate 18 loci in the isolate is sequenced. Then the sequence polymorphism at a single base pair level is decoded from the data. Okay. And two independent visualization tool we have used. One is the classical phylogenetic tool that gives you the phylogenetic tree in the form of clusters. And in another parallel approach, here comes, I already told you, we borrowed the technology from medical microbiology, converting the sequence polymorphism into something called allele numbers, right. So, the unique numbers were given to each one of those variations, and in the end of the day we created a bunch of allele numbers for those variations. It is a huge matrix of 18 on one side the loci and 24 on other side the isolates. So, it is a full of numbers, and those numbers were taken on board for another kind of population genetic analysis that also gives you the genetic similarity among the isolates. It goes like this: if all the alleles are two isolates, they share all the alleles. We hypothesize that these two are duplicates. You got it? If there is a variation, then that depends upon the number of variation. We started calling them, okay, one evolved from another one, or they shared a common ancestor. The story goes like this. I think in picture I will tell you how it is really done. This is something like this. The variation at single nucleotide level is converted into allele numbers, right, and the string of allele numbers is unique for each isolate, right. String of allele number means we decide in which order the loci should come. 18 loci are there, and the allele numbers are assigned for each and every loci, and it is end of the day it is what you call a long number put together. It is a string of allele number. In molecular microbiology, this particular string of allele number is called sequence type. It is a very unique one. This is something like the sequence types are the one which is characterized at genome level, right. This particular technology is something parallel to the whole genome sequencing technology, right? You hand pick the genes which is scattered in the genome and look at the variability or variations at single nucleotide level, and the resolution of resolving potential of this particular approach is as good as the whole genome. So, this is a power. So, in one side you could have sequenced the 24 strains. So, we did not opt for that because considering the time and money. So, you do only the handful of genes on board, and yet you get the same information we wanted. That is what exactly we did, right. This is what we call the amplicon of all those genes, and this is each gene is specifically curated and trimmed, right, and annotated and uploaded in GenBank. So, you will find something around 264 housekeeping genes representing around 24 isolates, right, and this is the virulence genes, again 168 is a huge matrix, right. And now you come to the next important. So, what you do now, you have the information of 18 loci from 24 isolates. So, what you do, you can create a kind of a large sequence length by catenation. This is popularly called concatenation, right. Here you just stitch all the sequences in a predetermined order and create a larger chunk of reads. Okay. Of course, everything has to undergo proper sequence handling. You have to look at the trace file, do the end trimming, do the quality check, and come out with the good base calls that you elevate into the analysis. There should not be any sequencing error coming into your analysis. You have to be very careful what is sequencing error and what is a genuine mutation coming from nature, right. So, if sequencing error comes from the machine, you cannot score it for mutations, very, very important point. So, once it is stitched together, then you subject the analysis into a molecular phylogenetic tree. This is what we got, and this is something like this. We could see around 3 clusters within the rice population itself, right. This is 1, 2 and 3, and expectedly the finger millet and pearl millet, they stood apart, right. It is coming somewhere here and this is somewhere here, and the message here is again the one which is causing blast in finger millet is slightly closer to the rice as compared to the finger millet. Finger millet is stand apart. The strain which is causing finger millet is completely different from the one which is causing rice blast and also finger millet blast. So, the new approach I already highlighted here, this is going something like this. So, we have given the allele number 1 to the first strain that was sequenced in 2004. So, that is a starting point, because prior to that nothing was there. So, whatever read was there in 7015, we just string of ones, right. It is 1 1 1 1 for all those 18 loci, right. So, that becomes a reference for us. With reference to that genome, how other genomes are playing, right. The surprising thing was the Indian strain which was sequenced by our effort, out of 18 there is only one difference in one of the loci, that happened to be a loci which is governing virulence function. None of the housekeeping function is different. That is expected also, right. So, here it is 10 and here it is 1 MPG2 loci. It is not same as 7015. That makes what is 7015 and what is RMGDL. So, that was the difference, right. And when we elevated to, I just highlighting the representative, the one from rice, one from finger millet and field millet, another one from finger millet. You can see for yourself. For example, I just draw your attention to MPG1 again. The allele 10 is very unique, no matter whether it is a rice strain or PL millet strain or finger millet. Good information for us. And second thing, the allelic profile is completely different, right. What is there in rice, you can see here, what is there in fuel millet is completely different. If you stitch all of them together, create a code, this code is different and this code is different. This is something like molecular barcoding using multi-locus sequence analysis, right. So, this is what you call the data set. Each number represents there is a unique single nucleotide polymorphism in those housekeeping genes or effector genes or virulence genes, right. So, now let us see how many alleles are common. That is also very important for us, and how many alleles are different. So, if you see here, rice and pearl millet isolate, they shared only 6 of the 18 alleles. That means 12 of the alleles are different between rice and pearl millet. Okay. When you talk about rice and finger millet, they shared 11 of the 18 alleles. So, what does it convey? The previous molecular phylogenetic tree also I just highlighted that finger millet is a little bit closer to rice. So, that is also once again reflected here. The 11 of the alleles is common between finger millet and rice. And the finger millet and pearl millet, only 5 of the 18 alleles are common, right. So, this kind of information is very useful in population genetic analysis, right, that speaks about the behavior of the pathogen population in a field or in a location, right. And the question here is how to visualize this result. That is very, very important, right. So, in a conventional what do you call molecular phylogeny, what you do, you have the sequence stitched together and you put it in mega 6 or mega 7 or mega 10, and you run the program that gives you a tree that can be easily interpreted by many. But when you have a data like this, it's a numerical number, it's a matrix, right, and a huge matrix, right, 24 and 18 matrix. So, for that only we have wonderful algorithms have been written in the past. This is one such algorithm. If you never come across, please note it down. This is what is called E-bust algorithm. This E-bust algorithm, it accepts the data coming from the what do you call allele data, and you can it accepts the allele data, and end of the day when you run the program, that is going to give you something like this, right. So, this tree is so important for us. It is something like the isolates, they share most of the alleles, they tend to cluster together, right. It is something like the birds of the same feather, they flocks together, same, no difference, right. If some strains are completely different from each other, then they will take a singleton position in the population, right. So, this is something like in a you will when you do this analysis, you will come to know whether the population you are handling is a clonal or it is non-clonal. That is number one information, right. If it is clonal, and you should be alarmed, because clonal populations are so dangerous in agriculture, it can create havocs, right. If it is non-clonal, nothing to be alarmed, because there will be natural checks, which is normally it is happening. So, this study through E-bust will reveal the population behavior, especially the pathogens like Magnaporthe, Ralstonia, or the clonal population, Xanthomonas in some cases, is very, very useful, right. A little bit story on that. E-bust V3 is a program, and a lot of variants are there. This is a website where you can download all those things. Of course, I already highlighted the key thing. There is 3 distinct population. They are quite different at genetic level is there, and there is an intra-population variation was there. And one point I forgot to mention here, the previous tree, whether you noted or not, the neck blast and the leaf blast isolates are typically one and the same. There is absolutely no variations what we found in 18 loci. They are same, not even a single base pair variation we could see. So, that again says that it is the same pathogen which is responsible for 2 different stages of blast in rice. So, just to highlight that, somewhere here, where is LB is a leaf blast and NB is an egg blast. So, they are one and the same in the same cluster, no variations. If there is a variation, then it would have branched out, and absolutely no variation for at least 18 loci, right. So, I will just take it forward. Now, the question is, we found 3 broad clusters, and from allele information also we could see some kind of concordance is emerging. Does it really reflect in the actual phenotyping? That is a big question. So, my colleague Dr. Prakash is working on that. So, what we found, we took the representative isolates and we started working on host range experiments, right. How does they really behave? Genetically, we found that these 3 are distinct population from each other at genome level, right, at least with the help of 18 loci. When this population is inoculated on the respective host, and we have taken some common host like wheat, oat and barley, right. This work was done, and if you see, we found that initially the rice isolates are capable of infecting only rice, and finger millet isolate is capable of infecting only finger millet, and pearl millet is capable of infecting only pearl millet, and the red one, they could not cross the barrier. Rice never infected finger millet and pearl millet and vice versa. The most interesting thing is the common host what we have taken on board, random selection, we have not taken all the cereals, but some random selection, wheat, oat and barley. These 3 strains are capable of infecting this common host. That is very interesting, right. So, this also reveals a lot about the behavior of these strains, right. And these 3 isolates, they are independently capable of infecting, apart from their main host, a common host like wheat, oat and barley, but they never infected rice and finger millet in the case of pearl millet, right. This is again aligning with the genome data whatever we generated from our molecular. So, this is a pictorial depiction, I just skip this one. Yeah. So, there is a kind of corroboration here between the genome data and also the infectivity assay data. So, that reflect the behavior of this particular strain. I will take another few slides just to tell you can we use this information to develop some kind of diagnostics. Before that, did you get some information about this utilization of genome data to address some biological questions, right? The key take home message here is you do not have to resequence every strain. You can alternative technologies are available. One such technology is you just handpick the genomic region, design primers, and you do sequencing, and under a technology what is called as multi-locus sequence typing or multi-locus sequence analysis, right. Multi is a word, you can take anything more than 2 is multi, and larger the number it is always better the resolution, and again you have to optimize how many minimum should be there. That is very, very important. You cannot have an arbitrary number. 18 we have taken. Again, anybody can question why 18, right? All housekeeping genes are common. You narrow down that. So, that optimization you have to do it. I cannot pinpoint what should be the number, right? But at international level, the scientific community has agreed for a number. That number is 7. Right? That means you have to handle minimum of 7 loci information for any multi-locus sequence typing, and the algorithms have been written only to accommodate 7, not less than 7, right. 7 and above they can accommodate, right. That means you need the information of at least 7 loci to make any kind of interpretation from population genetic point of view, right. So, with that, if that is clear to you, this is the rest is very simple. We found variation among rice, finger millet and pearl millet, and that variation is converted into a kind of a specific marker. That marker is something, this is based on some bioinformatic flow. We finally narrow down to couple of loci. It is having the discriminatory potential among the 3 strains, and to start with we have a PCR based diagnostic tool for pearl millet infecting Magnaporthe. That means they have the ability to discriminate the other 2 and the pearl millet. So, that is the status what we have right now. And just for the benefit of some students, what other information the pathogenomics can offer, which can help the scientist who is working in crop improvement programs. This is one program which is really having a lot of potential, what is called as effector mining from genome sequence data. So, PHI database, which I already told, there is only one database I told, there are a number of other databases have come now. You can identify the potential effector coding regions from the genome data, and I think all of you must be knowing effectors are pathogen encoded proteins. They are by and large coded by AVR genes, and the pathogen evolution into new variant is largely depends on the mutations in these AVR genes. That means the mutated effector can knock down the so called origins, right. So, already a number of examples are there in the literature, some of them I already highlighted here. And again, with the help of these effector coding genes, you can get into interactome studies with rice, and you can clone the so-called interacting proteins or interacting genes from rice. Those genes are what is called as the receptor genes, right? Popularly called as OR genes, or also called as cytoplasmic receptor genes. And this is also called as NLR, that is nucleotide binding domain, and leucine rich repeat region containing proteins. This is another benefit what you can harvest from this kind of programs, right? Of course, the interplay between effector and host targets and the matching immune receptors is shaped by intricate molecular mechanisms and exceptionally dynamic coevolution. This is ongoing, we call the battle between these two. This is what is called a genomic assisted elucidation of host pathogen interaction, accelerated in the genomic era. Now you will find more and more data about the so-called receptors. Now it is cloned now, several of them, and effectors have been cloned in many Magnaporthe system, and the interactome studies are now all the time published by many workers, right? And this is some of the wishful thinking from pathology point of view. The conversion of host genome sequence, pathogen genome sequence, and the host pathogen interactome would aid in the identification of host receptors or NLR or OR genes. That's going to be the long-term objective of similar kind of projects. And we have also the molecular docking has really evolved to understand the interaction between two proteins, right? The docking studies, that also would be very handy here if you have the capabilities of understanding the protein of those sequenced pathogen genes or sequence the host receptor. A lot of studies possible using these molecular studies, molecular docking studies. In our own study, we found around 30 effectors from rice, and overall we have around 40 AVR genes have been identified in rice Magnaporthe interaction, and some of them are just highlighted here, right? And any pathogenomic study, we anticipate certain outcomes, and I just put it in, it's my thought process here. First in the list is, of course, I already told the transboundary movement of pathogens. Right? Field pathogenomics would really help us, right? Transnational and interstate movement of pathogens, pathogen monitoring and molecular epidemiology is one field which is going to be benefited out of the pathogenomics projects. And we have already witnessed in the case of what you call Ebola outbreak, or it is in the case of blast outbreak in Bangladesh, it was a reality. The data was instantly uploaded. They created an open wheat blast portal. Anybody can access that, and anybody can host also their information. And this sharing of knowledge and information helped people to understand from where the pathogen has entered into that particular country. And of course, diversity within the species is another big question we always ask, because many times new origin containing cultivars knocked down by new variants. To understand what is that variant, where exactly the mutation is there, we need the pathogenomic approaches. The diversity within the species can be explained precisely. This is what is called as molecular diversity and strain identity. It's very new host adaptation can be explained by genome variability, that is identification of new entries in the population. Again, it's possible through genome data. And high sensitive and selective targets for diagnostics, we already shared. We could develop paired millet infecting Magnaporthe diagnostics using a couple of markers. And of course, effector mining from the pathogen genome can help us indirectly to fish out the receptors or receptor containing coding genes from rice. That would help us in the long way in crop improvement programs. I think this will be the last slide. And the team who contributed in the work, I was heading this project, and I was supported by my RA Neelam and also Dr. Prakash, my colleague. And we thank many funding partners here, in particular CRP Genomics, NAHEP, who supported this project in a big way. So thank you all, and if you have any specific questions, let's have some interaction. I think coordinator would give platform for this interaction.
Sir, for receptor mining, only in silico analysis are done or any in vivo studies are done or something?
It starts from in vitro definitely, and finally we have to come to in plantar level. So, so far such a study, you have two broad approaches. One is forward genetic screen and reverse genetic screen. So, reverse genetic screen, it is yet to make any dent in this program, but forward genetic screen is already going on. Your rice transcriptome data can be the starting point.
Sir, how whole genome based molecular phylogeny is done?
Whole genome based molecular phylogeny, I already explained here. If you have the whole genome of number of entries, you put all of them together in the what is called as alignment, and based on the alignment you create a tree. You are not taking one gene here, rather you are taking entire genome. And one point I must tell here, the gaps are ignored here, please. Okay, many times the gaps will be there, that should not reflect in the form of variability. So, the gaps are ignored and you put the entire genome sequence in the molecular phylogeny. You got it or not? Yeah.
Sir, we have the host genome and we have also the pathogen genome. Can we derive anything in bioinformatics or in silico analysis for this?
Directly putting them together, I don't think it is possible right now, but what is real here is if you can annotate the specific virulence protein, right? From that you can, if you can annotate your rice genome also, and we know that most of the R genes are NBS LRR type, right? You take them on board and take this effector also on board, you do kind of protein modeling, and you can get into this molecular docking. That is one thing I have in my mind. So, we are also planning to do in our project, just to know which two proteins are having high degree of interaction. That is what we want to know, right? The one which is having high degree of interaction, that means they really interact at a cellular level and moderate other cellular responses in rice, primarily activate the so called effector triggered immunity in rice, that is perceived as HR. What we call those varieties are resistant varieties. It is possible. For that you need to have a specific annotation from pathogen genome and also specific annotation from the plant genome. So, come to the protein level, and this modeling docking studies are now many people are doing this docking, especially the pharma, they are doing routinely, and I also had an experience in our DBT project we could do. So, it is, algorithms are already there, it is possible, yeah.
Sir, actually I am working on permanent blast for mapping of the gene for that.
Okay, very good.
For my curiosity, I blasted, I took many of the rice genes which are sequenced and I blasted in the permanent genome. Fortunately, I got one PI25 gene from the rice, which is query cover almost 99.9% means in that, and coverage is also 100%. But unfortunately, I didn't got the linked gene there. It was on the chromosome 1,
okay.
the PI25 related this one, but I got the linked gene on the 4th chromosome of the resistant gene on the 4th chromosome.
Okay.
What might be the reason?
That's what I'm telling you. See, I didn't bring all those slides. We did a comparative genome analysis of this rice and pale millet. What we found is only few genes are different, right? And those few genes present in those pathogens are completely different from each other. So, their interacting protein is completely different. That's what I believe, right? We found only 4 genes which is having virulence function is unique in those what do you call respective pathogens, right? And definitely if it is completely absent in rice, which is present in pale millet strain, and vice versa. So, that means the interacting protein may not be there in the respective host. So, that may not be the right approach. You need to change, because now pale millet genome is already there. More than hundreds of genomes are there, right? Pale millet genome pathogen genome is already there. It's available in public domain. You can handle this genome now. Yes.
Sir, what you said that PM permanent Magnaporthe deadly isolate, that's its sequence. How many virulent genes are there?
I told you, around 849 genes we could annotate using PHI database. So we believe most those genes are having direct or indirect role. Direct role in terms of effector, it may be less than 100, that we have not yet done that. So in rice we found around 40. So I think almost same number or some plus minus this way, that way you put, that we have not yet annotated. Yeah. You look at the effector coding genes, that would give you much better information than other genes. They play a supplementary role in pathogenesis, because in the pathogenesis, if you look at starting from spore landing, germination, finding the weak spot and penetrating, and beyond that it enters into the host system, number of genes are involved, and the expression of those effector genes are typically temporal. It's not expressed all the time. You understand? So during germination certain genes are expressed, post germination those genes are even repressed, and the product of one particular effector gene can play the starting point for expression of another effector gene. So it is a typically what do you call a temporal spatio-temporal expression that you need to address. Otherwise, if you go for a blanket effector expression, then you may not get the right one. Okay.
So in this Augustus, can we use for the higher ICAR, so levels and this?
I cannot answer this question, but there are so many other gene prediction softwares are there.
Yes, sir. I am using the FGenus.
FGenus is there. Yeah. I cannot answer this question. What we handle is a small microbial genome. We got the results, but higher, I think you need to approach.
So what is the next step after the gene prediction? How
Transcriptome you do and validate that.
Okay.
This is only a predicted CDS. They may not be true. So remember, your whole genome need to be validated using your transcriptome. You take 2 different or 3 stages of your host, generate RNA-seq data, and you validate your CDS, whatever you annotated, using the transcriptome data. And do one-step validation using qPCR also, just to have a confidence on the data whatever generated. We have to do. It's integrated in any genome sequencing project now, right? Yeah.
Thank you, sir.
Yeah. Backbenches, only front benches are asking questions. That's what I was wondering. Okay, fine. If no more questions, I think I thank the organizers to share our experience on this very interesting pathogen and challenging pathogen. And if you have any more questions, you can either contact me directly or through your coordinator. You are most welcome. Okay, thank you one and all. Yeah.
On behalf of the students and also the organizers, sir, I would like to thank you for sparing your time. I know you are busy.
No. We are available for this project only.
From